Great. And I will turn over to Jeff to introduce our next speaker. Jeff, please.
It's my pleasure to introduce the next speaker, Yu Gao, Tom. Tom received his Bachelor in Science from the highly prestigious Peking University, and then did his PhD and postdoc at the Scripps Research Institute, first earning his PhD from Thomas Kodak's lab, and then completing his postdoc with John Yates. And that's actually where I had met Tom, in the Yates laboratory. And one memory I remember of Tom was that John was quite excited because Tom had actually broken the code to unlock the thermo raw file. And this was actually just like a funny moment. And John was very excited, but Tom was like, played it quite cool, like it was a minor thing. And this really gets at Tom's his niche. He's very uniquely positioned between an interest in analytical chemistry, proteomics, chemical biology, but he's also very efficient on the informatics side. And more recently, he's gotten really into some cool applications of single cell proteomics. And I think Tom's going to tell us about that today. Thanks, Tom.
Thank you very much, Jeff, for the introduction. And let me see if I can share my screen successfully here. Can you see my screen?
Perfect. Yes.
Okay. Yeah. All right. Thank you very much, Jeff, for the introduction. And yeah, so when I first arrived, I didn't know much about proteomics. And I want to thank John for accepting me as a chemical biologist into his lab, and basically learn proteomics from there. Yes, I remember, like, when I first joined John's lab, Jeff is about to leave and start his own lab in Northwestern. And it's just, life is full of coincidences. 4 years later, I joined UIC in the same city. So today I'm actually very happy to actually to do this encore of the presentation I did on the US HUPO, titled like Automated Containerless Cell Processing Method for Single Cell Proteomics. So yeah, my lab here at UIC, University of Illinois Chicago, has been focused on single cell proteomics, and for the past like 3 to 4 years. And we have actually developed a different method. And today I'm going to talk about one of the method I think is one of the most significant improvement that we actually discovered is through a better sample preparation in single cell. So first, let me start with like why we do single cell. The reason, why we do single cell is really not just because it's a single cell, it's a hot term or anything. I think the real reason is really because we all want to do a highly sensitive proteomics. Basically, there are larger cells, there are smaller cells. Even in the single cell field, there's actually larger single cell, smaller single cell. But I think it doesn't actually, matter that much. It really, like, as long as we are pushing together, the detection limit of current proteomics method, and then trying to use this highly sensitive method to decode some of the cell heterogeneity, either in tissue or in cell culture or in tumor or the brain in neuroscience. I think that's the goal of the single cell proteomics. At least that's the goal of our lab. So here's the overview of the challenges of single cell proteomics. So first, we always start with a single cell, and then we do the lysis. After we do the lysis, we have the proteins, then we can do reduction alkylation. This step is optional. And then we use trypsin to digest them and do the typical LCMS separation. So there are, all of these different processes, and we all know that protein cannot be amplified, at least not now. So we don't actually have a PCR for protein. So because of this, we have to work with what we have. And so I think the first challenge that we identified is that there's simply not enough sample because we cannot amplify protein. The second is, like, if we work with just this much of protein, can we actually ionize more? Because we all know that we are not actually ionizing all the peptides from the digestion. So lots of the peptides, they got suppressed, like either ion suppression or low ionization efficiency. So can we actually find a way of working with the existing method, existing amount of method, can we actually improve ionization to actually further improve the sensitivity of single cell proteomics? And then the third problem, the third challenge that we identified is that there's not enough signal. So even if we actually did pretty well on the ionization part, there's actually still a problem with the spectrum, mainly because mass spec by itself, there's actually the electronic noise, the DAC and ADC conversion inside that produce electronic noises. So the signal to noise ratio is not always great, especially when you have a very small amount of sample. The signal may look like noise, and how to differentiate these signal to noise, that's another challenge I think we can actually tackle. So our solution to, or our answer of these questions, the first one is, since we don't have enough sample, can we actually eliminate sample loss? So our solution is a device called LEFCell. So LEFCell is an acoustic levitation device that we developed, and as we claim, that's the first containerless cell processing system that's able to process the whole cell, including cell lysis, digestion, alkylation, and then even TMT labeling, and then inject everything into the mass spec, into the LC-MS system. So therefore, we lose less sample, so we have more sample preserved for the mass spec analysis. The second problem is not enough ion, and we developed some alkylation strategies. That's an extension of a previous paper published by Lloyd Smith Group a couple of years ago. So we demonstrated that these alkylation can actually enhance signal on the protein-wide level. So it's a global level ionization enhancement by chemical reactions. So the third one is we actually developed machine learning models basically to do noise reduction to improve the signal to noise ratio. Currently, we're working hard on improving this method so this model can actually be transformed. Currently, it only works on the machine that it was trained on. So it was trained with my machine, my QEHF. So currently, it works very good on the data that generated from my QEHF, but not necessarily on other machines. So we're trying to improve on this. So well here, so if you remove the noise, the signal will be much more obvious, and the signal to noise ratio will be higher. So here is the version 4 of our LEFCell platform, and that's will be the main focus of today's talk is the automated containerless cell processing system. So I'm going to talk more about the system in later slides. But before I actually start talking about this containerless system, I want to actually go a little bit into why we want to actually develop this containerless system. One of the main reasons is that when we're trying to do single cell proteomics, we find that many times we lose all of our sample. And then I start to question, can we actually use the plastic tubes? Are we actually using the right kind of tubes? Is there a better tube? Many tubes, actually, vendors claim that their tubes is low binding. They preserve more proteins, but, and is there actually the best tube for single cell proteomics or nanoproteomics? So we published this paper last year on JPR, that evaluation of the binding preference of the microtubes for nanoproteomic sample preparation. And what we find is when the sample goes down to 100 nanogram, about 1,000 times more than a single cell, so this represents about 1,000 single cells, then we start to see very significant differences between different tubes. So each of these bar in this bar graph, each of these bar actually represent one brand of the tube that we tested, and the number in the y-axis is the recovery percentage that we got after incubation. So as you can see, it's temperature dependent, but also time dependent, and the highest, the best tube can perform as much as 7 times better than the worst tubes tested. And that's a problem because when you actually see a published result and trying to reproduce some of these results, if you're not actually using the same exact type of tube, you may actually not be able to actually reproduce the result. And for single cell proteomics, this is even more troublesome. And the problem actually is even more severe than just losing peptides to the tube through this non-specific binding. We actually find that different tubes from different vendor actually binds to different peptides. So as you can see here, when we did the hack digest, the peptide that identified from different tubes, they actually possess different lengths when incubated with different brands of tubes. So in other words, your identification and the biological answer actually is highly dependent on which tube you actually use when the sample is small enough. This is not actually significant at all when you have a sample that's more than a microgram or 500 nanogram. It only becomes significant when you actually have a small size sample. And in single cell proteomics, and of course, we have a small size sample. So the current way of actually reducing this phenomenon, like a protein peptide surface binding, is actually mostly focused on shrinking the size of the droplet. So here are some of the solutions from previous labs, including the silicon wafer using photolithography, like the nanopods published by Ing Zhu and Ryan Kelly in 2018. And then the more recent, like fluorocarbon coated glass and pop from the Nicolas Lavos lab. So they all are focused on shrinking the size of the droplet into a nanoliter size. However, when you shrink this into a nanoliter size, there's still a couple of problems. First of all, there's the evaporation problem that you need to actually control the humidity problem. But the other thing is this is not actually easily accessible. So these things need specialized equipment and also specialized optimization for these single cell work. But even on top of that, this does not completely eliminate the surface. So in other words, yes, the surface contact is actually small, but you're still actually losing some of the peptides and proteins to the surface. And that's kind of unavoidable, even with like fluorocarbon coating, because some peptides are hydrophobic, some peptides are hydrophilic, and for like more sensitive detection, we think a better way to do that is just to eliminate the container or the slide completely. So that will completely remove the peptide to surface interaction. So basically, there's no liquid-liquid or liquid-solid interface. There's only a liquid-air interface, which the peptide won't actually go out to the air and lose the sample from there. So here's our solution. And if you haven't actually seen it, I encourage you to actually go to our website, cell.lab.gy, to actually see this in action. So we claim this is to be the world's first containerless cell processing system. It uses acoustic levitation and to levitate the single or multiple cells. We actually levitate tissue as well. For example, a small organoid can be levitated with this our device. So over the past year or so, we have improved our system. So now the system includes automatic cell dispensing system, lysis, digestion, injection. There's no liquid transfer steps in the middle. So we also have an imaging readout, multiple measures with minimal sample loss. And we can also do DNA, RNA extraction, which is crucial to multiomics measurement for a single cell. Here, I'm just showing you a video showing that we levitate a droplet with a fish egg. So we actually choose one of the fish egg from a sushi place. So the fish egg is big enough to see it, and also it's stained, so you can actually see the fish egg in here. If you watch this like long enough, you will actually see the cell will be actually lysed. And as you can see, we can actually move this cell freely up and down, the droplet in the middle of the air without any support and without any touching any surface, right? So this is actually acoustic power. So how does this work? So acoustic levitation is not actually a stranger to mass spec. It works as two or even one ultrasonic transducers produce a sound wave. And when the two transducers are perfectly aligned, they create these like low pressure zones and high pressure zones called node and antinode. So in the low pressure zone, we can actually put the levitated objects in these nodes areas, because these are low pressure zones and they are supported by the high pressure zone, from above and below. So since like 2008, even before that, but some of the related work, like Lloyd Smith published this paper in 2008 using acoustic levitation to actually sample some peptide metabolites using this method called CALDI. So these are analytes directly dispensed into the droplet, and then they can be analyzed directly to the mass spec. Later, the Bushkamp lab and also Dietrich-Vollmer's lab published work also on using acoustic levitation to sampling peptides and other species with the mass spec. However, our claim is our system is actually the first one that does not actually need to actually put a purified protein or anything else. We can actually start directly from a tissue, a cell, or multiple cells. So we start with the cell processing in here. So the whole system is controlled by a system on a chip. So this one is a computer, and everything is actively controlled. Actively controlled means that we actually have sensor that actually can detect the humidity, the temperature, and also cameras to actually detect where our robotic arm is. So these can be have a feedback loop. So this computer can actually control what's the next step for everything. And we have a temperature and a humidity control. So we can do live cells. The cell is actually alive in the droplet, and the enzyme activity is also preserved. So these are the advantage of the system. We have the transducer array on the top and bottom, creating these node and antinode ultrasonic waves to levitate the cells. We can actually have multiple nodes, so we can place multiple cells at the same time. The cells are actually sorted by a microfluidic chip, which is commercially available, but also through collaboration with Ian Popowski's lab here at USC, we can actually deliver single cells into these automatically. So after processing the cell, we can, these robotic arms can actually add different reagent and enzymes to that to perform reduction, alkylation, digestion of the protein, and eventually suck the sample into the sample loop and then go through a C18 column and go to the LC-MS directly. So here I'm going to show you this system, the current version 5 in action. So here you can see that this robotic arm is actually dispensing these small droplets containing 1 cell per droplet into the air, and then it can actually add with contactless this reagent to each of the droplets. So these are just food color yellow, but in reality we can add reduction and alkylation reagent into this droplet. And we can even do TMT labeling with this. So as you can see, we suspended 5 cells simultaneously in here. And after digestion, a typical digestion takes 30 minutes or so, and we can actually inject them all in by this mechanism. And then we can start another run. This whole process is controlled and fully automatic, so you don't actually need any human involvement. So the system can run 24/7. Sometimes we do suffer from stability issues, but that's we are still improving that, and hopefully this can run multiple days and processing thousands of cells. Currently, we're at about 200 cells per day or so. Okay, so here are some of the key dimensions. So there's about 4 millimeters between each of the nodes, and our droplet is about 6 microliters. So that's not nanoliter dispensing. So we don't have to worry too much about evaporation, and we can actually grow this thing up to about 10 microliters or as small as 1 microliter. So this size is actually adjustable. We dispense these, reagent can be 1 microliters for reduction and 1 microliter for alkylation, and these reagent can be added contactless, so without actually cross-contamination between the nodes. So on top of that, our system can also perform RNA and DNA extraction. So here is actually a droplet full of magnetic beads. So we first deliver magnetic beads, which can capture RNA into the droplets. After lysis, these magnetic beads will capture the RNA and DNA inside. And then we use a robotic arm to actually, with a magnetic needle, to actually concentrate, as you can see here, in this this droplet, from a very cloudy droplet becomes a very clear droplet, because all the magnetic beads are being extracted by this little arm. And finally, we can actually leave it. So this way, we actually extracted all the magnetic beads. So you can see that on the tip of the needle is the black magnetic beads, which is extracted from the droplet, and the protein and metabolites are still in the beads. In the droplets, while the DNA and RNA being separated. And we can then use this to do proteomics and RNA-seq from the same cell. So here we're showing that our camera that's detecting the fluorescence detection of the cell. Each of these little shiny dots inside is one cell. We put multiple cells in this droplet, showing that that there's multiple cells inside. So these can be served as a platform to detect internal fluorescence or certain fluorescent assays. We can also use luminescence assays for this purpose. But here we're just showing the imaging capability that we have that can actually do phenotype screening of the cell before we actually do single cell proteomics or transcriptomics. So the system is quite versatile. So here are some data showing that what's the advantage of doing the single cell proteomics using the left cell platform. So as we can see here on the PSM level, peptide level, and protein level, the left cell processed 50 cells are significantly better than the PCR2 processed 50 cells. And if you see in these in the assay here, which is use the scope MS method, when we have one channel, which is the blue line, being the 200 cells, and then the other 4 lines being 250 cells, 250 cell processed with left cell, and 250 cell processed with the PCR2. And as you can see, when we filter them by the signal to noise ratio, the 50 cells processed by the PCR2 drop significantly, while the signals for the 50 cells from the levitation device preserve and as good as the 200 cell signals at the protein level. And these are the 50 cell tests. Here is the single cell test. So here's the protein comparison, peptide comparison, and the PSM comparison. So the levitation device constantly delivers 30 to 40% more IDs from each of the experiment. And these experiments are done without any match between runs. And these are done on our own Q-Exactive HF machine. So it's not a timsTOF HCP or SCP, it's just our QEHF. And we can do all of these with each of the single cell process with our single cell levitation device. So here is what we did is we sampled 50 single cells. So one cell at a time. We repeated 50 times with the levitation and the PCR. As you can see here, the relative cell intensity ratio is actually there's a very significant differences between the levitation and the PCR, showing that our device really preserved more samples. So in the future, this whole system actually to build the whole system actually costs less than $1,000 and can actually process more than 1,000 cells. That's our goal. Currently can process about 200 cells per day. But however, you can build 4 of them and you can actually use these in parallel. So they can do automatic multiplex labeling. That's TMT10, TMT16. Then you use a low flow, small diameter LC to deliver your sample to the LC-MS. They can do magnetic extractions to show that to actually obtain RNA-seq informations from the same cell, but also you can also do affinity purification MS, including phosphorus enrichment and other antibody-based enrichment method using magnetic beads, as we show that the extraction works. And then you can do fluorescent imaging, you can do phenotype screening through bright field or fluorescent imaging to see how do these work, right? So these are actually the power of the potential of our system. So in summary, our lab cell provides a containerless live cell processing solution. So you actually process live cell and process the cell through all the necessary steps to get the proteomics data. It reduces the lipid transfer and surface contact, it prevents sample loss, and its fully automated platform provides an affordable solution to high throughput single cell proteomics. So here's my email. If you have any question, feel free to send me an email. If you want to collaborate with us, also just please, send me an email. Anyone wants to join us to do a postdoc in the field of single cell proteomics, biology, and informatics, please let me know. And I want to, thank, first my lab. Without them, nothing is really possible. So these are my students, my postdoc, some of my former trainees as well. And then I want to thank our funding support from the NIH, Chicago Biomedical Consortium, the College of Pharmacy, and UIC, the NIH HUPO program, and the University Cancer Center and the Big Ten Cancer Consortium. And I thank you for your attention, and I would like to answer any question that you may have.
Thanks so much. Yeah, that's wonderful. Great. I wonder if Jeff is there. There's Jeff. All right, I give it to you.
Tom, very cool. I'm really impressed by this Levis cell thing. This is actually super neat. And it's quite frequent that people ask me, oh, what is the state of the art of single cell proteomics or single neuron proteomics? And I often tell them to check out your website and your paper. So that was
Thank you very much,
a great
Jeff.
talk. Thank you. I have a silly, naive question. What was the best tube?
What's the best tube? Yeah, so that's actually, we find actually, it's in our paper, but Appendorf, low bind, and the Sarstedt, these are actually the 2 best performing tubes that we tested. And they all use a technology called copolymer. They didn't actually publish any details about that, but they all say that these are copolymer tubes. And one is from Appendorf, one is from this company called Sarstedt.
Thanks, Tom.
Yeah, no problem. Well, I guess I presented like too many times and answered all the questions.
Yeah,
Tom, thank you.
Yeah, thanks so much. This was great.
Thank you for having me.
Technology. Yeah,
Thank you for having me.
of course. Thank you. Thanks, Jeff. Yeah, so